Yeah, can we start the presentation? Yeah, I'm audible, students?
Yes, ma'am, you're audible.
Yeah,
Yes, ma'am.
we can start. Yeah, we can start, I guess.
Okay, ma'am. Okay, ma'am, am I audible?
Yeah, you're audible.
Okay. Good afternoon, everyone. Today, Rupal, Shivani, and I are presenting on wilt of tomato disease, which is caused by Fusarium oxysporum. Next slide, please. Okay, so I'll start with introduction part. So tomato Lycoposis esculentum is one of the widely grown vegetable worldwide, as we all know, because a tomato is something we will find in every house, and it is widely used even as an unprocessed food or as processed food also. This wilt of tomato is caused by Fusarium oxysporum, and subspecies Lycoposis is the significant contributing pathogen of tomato vascular wilt. Fusarium oxysporum Lycoposis mainly causes wilt on tomatoes. So wilt is a symptom which is caused by lack of water, which shows that the plant is drying out, and uh, it doesn't show plant as very it shows the lack of turgor in plants, and soon plant dries out, and it soon dies. But it is mainly caused in tomatoes, but it can also infect many other plants including potatoes, peppers, eggplants, and legumes. The initial symptoms of the disease appear in the low leaves gradually, but then it causes wilting of the whole plant. The disease develops more quickly in soil that are high in nitrogen and low in potassium, because what happens is tomato need potassium in high amount as compared to other nutrients. So in soils which are low in potassium, there are more chances of the development of this disease. Next slide, please. Okay, now coming to the classification of this. So this causative agent belongs to the kingdom fungi. Division is Escomycota. Class is Sordiomycetes. Order is Hypocreas. Family is Nectaceae. Genus is Fusarium. Species is Oxysporum. And subspecies Lycoposis. So FSP Lycoposis means basically it means formal species. So it is given it is just an informal way to tell that a pathogen is causing a disease. So we just give this after species name to make things easier. Next slide, please. Okay, coming to the history, Fusarium oxysporus was the name was given by Cinder and Hans, which is a soil-borne plant pathogen in class Hypomycetes, and it causes Fusarium wilt, especially in tomato. This disease was first described by G. Mace in England in 1895, which it is worldwide importance, where at least 32 countries had reported the disease. At one time, the disease nearly destroyed potato production in parts of Florida and southeastern states of United States. And also, it had become the most serious disease of tomato in Baja California Peninsula. So here is an article which was published in 2005, saying that in Baja California, which is in Mexico, this disease was highly spreading, and after looking at the symptoms, it was characterized that it is caused by Fusarium species, and said that it was highly dangerous, and it affected many parts of the Mexico. Next slide, please. Okay, coming to the agent causative pathogen, Fusarium oxysporus. Lycoposis is one of the most destructive necrotropic pathogen, affecting crop crops, causing considerable field and greenhouse yield losses. So it also it is caused in field also, and greenhouse yield losses. Greenhouse means if you are growing the tomatoes even in a controlled environment, all is very controlled. Even then also it is caused, and it results in greenhouse yield loss. It affects greenhouse and field grown tomatoes and warm vegetable production areas. So mainly warm areas are affected more. The disease is characterized by yellowed leaves and wilted plants with minimal or absent crop yield. So if this disease is caused, then crop yield is very reduced to almost zero, or it is almost absent. The pathogen invades the root epidermis and extends into the vascular tissue, and then it colonizes xylem vessels producing mycelium and cornelia. So due to this, a lack of water is due to this lack of water is seen, and the wilting of crops tomato plants is seen, as you can also see in the picture given here. Next slide, please. Okay, so now I would like to hand over it to Shivani.
So, life cycle of Fusarium oxysporum F.sp. Lycoposis. Next slide, please. The disease cycle is divided into several steps. Like here in the image, it is given with alphabets like A, B, C, D, E, and F. The step A is penetration step. This is the step when fungus penetrates and enters inside the plant. Different types of spores are present in soil. It can be microconidia, macroconidia, or Clamidospores, which germinates as a small hyphae. This small hyphae enters into plant by root hair, but this is not the only way of penetration of hyphae. It can also be caused due to wounds present on plant or by cracks formed during lateral root formation. Like here, in this cycle, the germination of Clamidospores in response to host exudates and direct penetration at root tip occurs. Step B. Germinating spore invades root cortex. Once the hyphae has entered into root hair, now it invades in cortex. That is, it grows on root hair and eventually reaches the cortex. After cortex, it spreads within the cortex and reaches endodermis, then to pericycle and ultimately to xylem vessels. Step C. Now the hyphae starts spreading within xylem vessels through pits. The pits is basically the gaps within the vessel, and it is seen that mycelium mostly moves in upward direction, that is towards the apex of plant. Along with spreading, it also branches and forms small spores called conidia. It can be macroconidia or microconidia. This conidia ultimately gets detached and moves in upward direction along with xylem sap coming from the roots. Xylem sap is basically the mixture of water, nutrients coming from the root, and these xylem sap reaches all parts of body along with the spores present, and that spore starts invading the other xylem vessels by germinating and leads to infection of many xylem vessels. Now soon the plant detects the pathogen that a pathogen is present and tries to stop the spread of this fungal hyphae. Fungal mycelium spreads in the xylem vessels, plant secreted antifungal compounds, and fungal mycelium clogs the vessels. How so? For doing so, certain cells of xylem vessels extends and forms moon-like outgrowth by overgrowing called tylosin, and starts to protrude towards the xylem vessel, and also some gums are secreted, and these tylosin along with gums along with fungal mycelium ultimately clogs the vessels, and because of this blockage mycelium won't be able to move through the xylem. Step D. Collapsed xylem vessels resulting in browning of vasculature. Lower stems begin to wither. How? As the blockage of mycelium won't be able to as there is a blockage, so mycelium won't be able to travel through xylem, but at the same time the uptake and translocation of water and nutrients stops as well, and without translocation the plants gets deficient of nutrients and starts to wilt, because the water or nutrients is not reaching to that particular part of plant, but the upper part of plant are still transpiring and all other processes are running normally. Step E. Infected plant witnesses complete chlorosis and withering. How? When one at a point is reached when rate of transpiration is very high with respect to water reaching over, then at this point, stomata starts to get closed as the amount of water becomes very deficient, and eventually photosynthesis gets affected. And due to all these, the plant starts to droop and gets deficient to nutrients, and eventually infected plant witnesses complete chlorosis and withering. Step F. Wilting and plant death occurs, and then there is a high chance that the leaf falling from wilting plant is infected or has spores on it, and when the leaf falls on soil, the soil gets infected, and hence by spore spread the new cycle of diseases begins. These spores can be of three types. It can be microconidia, which are very small colorless spores and are generally one-celled. Macroconidia, these are larger than microconidia, colorless, and are slightly curved and contains up to 6 cells. Then third is Clamidospores. Clamidospores are roundish, thick-walled, and may form in or on the mycelium. Next slide, please. Mode of spread. The fungus basically prefers warm weather like hot climate and acidic pH and sandy soils. The fungus is also seed-borne, that is live and survive within the seed, and can also be soil-borne, that is live and survive within the soil. Either it can be a spore form or can be of mycelium form, and can be carried away to long distances by biotic and abiotic agents. The optimum temperature has found to be 26 to 32 degrees Celsius when the fungus is most active, and low nitrogen, low phosphorus, and high potassium contents that are of soil favors the growth of fungi, and the spread can also be from soil as the soil becomes infected by planting infecting transplants, and from movement of infected soil, either it can be by water erosion or farm implement. Next slide, please. Here, I have attached a research article on tomato fusarium wilt. So, fusarium wilt of tomato has been reported in India since late 1960s, but so far there is no comprehensive study on the genetic diversity of pathogen which occurs almost in all production areas of country. Now the question is, why must we have to study these genetic variabilities, or what's so good about that? So, genetic variability of members of genus Fusarium increases difficulties encountered in development of resistance host genotypes, as well as is effectively deploying available tolerant cultivars. Such information may help understand virulence dynamics of pathogen and facilitates development of efficient disease management strategies using based molecular markers. The article consists of molecular characterization, pathogenicity and toxigenicity of F.oxysporum strains that originated in tomato growing area in 5 states of South India. These are Karnataka, Orissa, Maharashtra, Tamil Nadu, and Andhra Pradesh. The wilt infected plants and rhizosphere soil samples from 5 different states were collected, and total of 72 samples from different locations were taken into account. In the view of economic loss that is caused by fusarium wilt of tomato that Rupal will be further explaining, the objective of the report was to investigate the molecular characterization of pathogenic Fusarium oxysporum and non-pathogenic Fusarium oxysporum strains isolated from different growing regions of South India through various assays. And next slide, please. These are the final results and techniques applied during the research. The first is morphological identification, which basically revealed that all the isolates displayed morphology typical to Fusarium oxysporum, and mycelia of isolates appeared delicate, white to pink in color, or often purple tinge, and were sparse to abundant, and also there were all the 3 types of spores present, amongst which macroconidia sparse to abundant are borne on branched conidiospores or on surface of sporodokia, and are thin-walled, and microconidia were abundant borne on simple phyllites arising laterally, and Clamidospores, both smooth and rough-walled, were abundant and formed terminally or on an intercalary basis. Then DNA extraction of fungal strain was done. For DNA extraction, the strains were grown on potato dextrose broth at 28 degrees Celsius under static conditions. Mycelia were then harvested and lyophilized. Later, 100 mg of finely powdered mycelium was used for DNA extraction using HI-PUR A, which is a fungal DNA mini kit from High Media India, whose purpose is it allows rapid and reliable isolation of high quality total cellular DNA from a wide variety of fungal species and tissues. And the DNA sample were then stored at -80 degrees Celsius for further research. Species-specific PCR assay was used for specific identification of F.oxysporum. The PCR carried out by using the species-specific primer and 1 kb ladder was used as marker. In the PCR, the primers were amplified. The primer amplified a single 340 base pair DNA fragment specific to Fusarium oxysporum, which confirmed the species-specific identification. As you can see here in this picture, the M that is mentioned is the 1 kb DNA ladder, and from 1 to 17, these are the isolates of F. oxysporum. Amongst which the bright ones have the most number of strains of F. oxysporum. Next slide, please. Pathogenicity testing of strains were done to determine the forme specials. Virulence analysis of strains were carried out on a set of 5 tomato cultivars susceptible to Fusarium wilt. Tomato cultivars is basically a plant which is a product of intentional breeding. So here the tomato cultivars used are Arka Abha, Arka Lok, Arka Meghali, Arka Saurabh, Arka Vikas. These were the 5 tomato cultivars used. A total of 69 strains of F. oxysporum were tested for pathogenicity. 20-day-old healthy seedlings were inoculated by standard root dip method. Typical symptoms of wilt disease were first observed 15 to 20 days after inoculation. Symptoms will be explained in further slides in detail. The F. oxysporum isolates examined had difference in disease severity on test tomato varieties. Based on mean disease severity, MDS, the virulence of each isolate was recorded as low if the MDS was below 25%, moderate if the MDS was between 25 to 50%, or high if the moderate if MDS was higher than 50%. And the isolates were hence categorized as 31 strains were characterized as highly pathogenic, 20 strains were characterized as moderately pathogenic, 12 as weakly pathogenic, and 6 as non-pathogenic. And the uninoculated tomato seedlings showed no symptoms. And that's how, based on root tip inoculation test, isolates pathogenic to tomato were identified as F. oxysporum, and all isolates were successfully re-isolated from disease-infected plants, thereby completing Coates' postulate. As you can see here in this table, the first is strain number, then second is source, either it's stem or rhizosphere soil, then is geographical origin, from which state the sample was taken, then tomato cultivars for disease severity, then virulence grade, either it's high, moderate, low, or non-pathogenic, then it's GenBank accession number. Next slide, please. Detection and phylogeny of toxigenic F. oxysporum is done, and a total of 31 highly pathogenic strains were screened to detect and identify funonicin production. Primers were used for specific detection of potentially toxigenic F. oxysporum strains. When primer pair was used, single band have amplicon size of approximately 790 base pair was observed for fuminicin producing strains of F. oxysporum. Among the 31 selected strains of pathogenic F. oxysporum, 11 isolates were found to be toxigenic, harboring the same primer gene that was present in primer. The causal agents isolated from symptomatic plants and soil samples was identified in this research based on morphological and molecular analysis and pathogenicity testing of 69 strains on 5 susceptible tomato varieties showed 49% of strains were highly virulent, whereas 30% were moderately virulent. Next slide, please. So Rupal will be discussing about signs and symptoms of wilt of tomato.
Yes. So next we have is sign and symptoms. So the first is wilting. So the first symptom, it appears when the fruit is begin to mature, and this symptom it goes often very unseen. Initially, it uh, initially it starts with a single leaf or shoot or near the top of the plant, but uh, it will recover at night when the temperature is low. But as the symptom it it um, it moves, the entire plant will wilt and will not recover. So such a symptom it occur when the weather is hot and the soil moisture is ample, and also watering does not watering does not make a difference here. And as the symptom moves on, the base of the plant may show brown canker, root rot, and also the cross section of the infected stem may show a brown spot. And also the wilted leaves, it will dry out and will fall off. And the next symptom we have is yellowing of leaf. So it is the most common symptom in any plant. So the lower leaf will be affected first, and it will start only from the one side. And as the wilting moves, it will eventually move up to the plant. So next we have is low development. So it is very typical. It is very small, tiny, or no fruit develop. So it mainly occurs due to the deficiency in nitrogen or phosphorus. And if the tomato is grown in poor soil, so fertilizers can also solve these uh, problem. So plant in poor soil, they are lacking in nutrient and they are unable to develop properly. Also, there is a one more issue, which is light issue. A lack of sun can also affect a tomato plant. So it needs at least 5 to 6 hours of sun to survive. And next we have is diagnosis. So the easy way, the easy way to diagnose is to cut a fresh stem at the base of the plant and is placed in the water. So it will show a stream of white slimy substance. So I have also attached a video for the better under, uh, understanding, and I hope this video is also working at your end and you can see uh, properly. So as you can see, this white slimy substance, this is the strong indicator that the bacterium it is present in the uh, vascular stem, and the interior of the stem will be light brown in the pith area. I'm also playing this video one more time so that you can see it more uh, properly. So next we have is economic loss. So due to the high temperature and humidity, Fusarium oxysporum can cause damage. So it is the soil-borne uh, pathogen that can cause um, wilting, and it is the only host. So they report around 80 to 90% of economic loss, mostly in the temperate areas. And next we have is management uh, practices. So there is no such proper treatment for Fusarium, and since it goes on in the soil for many years, it can be very hard to eradicate. So it is very difficult to manage once it is there in the field. However, there are no chemical control that can provide to be effective, but here are some measures to help you to control it. First is to remove the infected plant. So if the wilt it is present in the field, remove the infected plant immediately. So the plant can be placed in a garbage bag or another container so as not to spread the soil and bacteria to the other plants. So we need to pull up the infected plant and destroy it, but do not compost. And this practice can also decrease the chance of infection spread through contaminated tools, gloves, etc. Next is apply the lime to the soil. So to bring the pH up to the neutral range of about 6.5 to 7, uh, and we should also clean all the tools that may have come in contact with the infected soil. Next is avoid the over application of high nitrogen uh, fertilizer. So the high nitrogen and low potassium can also increase the openness to the fungus. So for this, we can also use soil tests to know about other nutrient deficiencies. Next is next is rotate the crop. So since the pathogen it can persist in the soil um, for many years, so we have to find another area to plant. So a 5 to 7 year of rotation is now being uh, used, but even that does not ensure that the infection will be entirely gone. So we can also use raised bed to improve it. Apart from these, we can also avoid the activity in the wet planting and the movement of wet soil from place to place by shoes or tools will spread the infection. And also, one more practice is that rain and overhead watering can also bring back the contaminated soil into the leaves and lead to the infection. So for this, we can do planting in the spring or fall. And next are the references. So these were the sites we used to get the information regarding this topic. Next is thank you.
Okay, so definitely a good presentation. Lot of improvements from the previous ones, like you included a lot of literature and that streaming part you showed. I mean, this is when we the first I remember when I was in the university and the first practical we were doing when our teacher he took us to the field. We got the sample and this streaming thing he showed us that time, and it was such a good feeling that time also that okay, now you can see something, right? So this streaming test is can be done in the field also if you have some just few things. So this is again used for the diagnostics for many pathogens. So this was good overall. It was a good presentation, good content, and a lot of research experiments you've included about how they did the pathogenicity tests also and number of varieties. All those things was good overall. I liked it, and it was well prepared. All of you good. So any questions from the other groups? Fusarium is Oxysporum is a very important pathogen. It causes wilt disease in number of the plants. In that way, it is actually very important, right? And wilting can lead to the death of the plants also. So losses caused by these pathogens are actually more, right? So farmers, for farmers, this is a troublesome pathogen, and they always look forward to avoid and do whatever they can do to manage this particular pathogen in the field, right? Yes, any questions from the other groups? So this was our last group, or do we have more? No, we have one more, right? Nandini wala group. Nandini, are you there? No, she's not there. Who else is in that group? We have Nandini, we have Kshitij and Janvi. Janvi, are you there? Yeah, am I audible or not?
Yes, ma'am, you're audible.
Okay. So next we are having on 24th, if I'm not wrong. We can postpone it also if you want, because 24th is ideally the last week, I guess. So we can do it earlier also, no problem, right? So no questions from the students, I guess. So anyways, it was a nice presentation. Thank you so much. Yes, Namrata.
Shivani, why we use potato dextrose broth and what are its benefits?
That is a good
So,
question.
the nutritional rich base from potato infusion encourage mold sporulation. That's why potato dextrose agar is used, and dextrose basically supports the general growth of microorganism. And agar is, as we all know, solidifying agent. And we can't the normal nutrient agar or the broth that we use is basically for cultivation of broad range of bacteria, whereas the potato dextrose is mainly preferred for using for isolation and growth of wide range of fungi.
Okay, fine.
Okay, okay.
So do we have Yuvraj? Yes, we have a Yuvraj.
Yes,
Okay, so
she has mentioned
yes,
on
Yuvraj.
the slide about the root tip method. So could she explain it once?
I didn't get you. What is your question, Yuvraj?
She explained. She
Ma'am, he asked about root dip method, I guess.
Yes,
Yes,
ma'am, exactly.
students. Yes, students who are going to explain this.
Okay, root dip method is basically a method for inoculation. Like in this particular research paper, the seedlings were uprooted and root apex was trimmed, and then it was submerged in conadial suspension of each strain for 30 minutes, and then the inoculated seedling was put it in a mini pot in controlled environment, and which contained sterilized soil and other equipments, and the optimum condition was provided for its growth.
Okay, fine. Any other question? No, I guess, right? Okay, so I guess we'll close here. We already have a class tomorrow, so we can see, since tomorrow we don't have any PPT, right? So, oh, tomorrow we have a face-to-face class, right? So we'll see what topic we can take up tomorrow, right? Okay, thank you so much. I'll close the meeting now.
Thank you, ma'am.
Thank
Thank
you,
you.
ma'am.